Immunity is the capacity of a host to resist infections. Human body is powerfully equipped with diverse mechanisms to prevent the occurrence of various diseases. The development of disease is an elaborate procedure known as the mechanism of pathogenesis. Any infectious agent can be stopped at several levels to restrict the establishment of a disease. Body starts the hurdles for a pathogen at the site of entry in the body. This segment is dealt by the components of innate immunity. The mechanisms involved in innate immunity are first line of host defense and second line of host defense. These are non-specific host defense mechanisms. They respond to all the coming pathogens in same manner and do not respond differently to different pathogens. If these mechanisms are unable to restrict infectious agents, they come across adaptive immunity or acquired immunity. Adaptive immunity, as the name suggests, is adapted by some process. Host's body comes in contact with the infectious agent and prepares to fight against it. It is also known as acquired immunity. This is acquired in the lifetime and not present by birth. Acquired immunity takes around a week to develop after the entry of pathogen inside the host's body. During this period, innate host defense takes care of the invaders. Once adaptive immunity develops, it remains for a long period of time. The mechanism used to develop adaptive immunity is specific host defense mechanism. This mechanism involves humoral immunity or antibody mediated immunity and cell mediated immunity. Production of antibodies provides humoral immunity and sensitized T cells provide cell mediated immunity. Humoral immunity takes care of infectious agents which are present outside the cells. For example, bacteria, viruses or toxins present in blood or body fluids, that is in case of extracellular parasitism. Cell mediated immunity works against intracellular parasitism where the infectious agent resides inside the cells. The important features of adaptive immunity are antigenic specificity, diversity, immunological memory and self and non-self recognition. First, we will talk about antigenic specificity. Components of adaptive immunity can recognize the foreign substances specifically. The immunity developed is precisely restricted for the agent which has provoked it. The response may be in the form of antibodies or sensitized T cells. The antibodies will be specific for the antigens which have stimulated their production and will not bind to other antigens. The sensitized T cells will recognize and destroy only the cells which have led to their sensitization and not the other ones. Next is diversity. Components of adaptive immunity can recognize numerous infectious agents and have the explicit capacity in differentiating between them. The immune response generated against different invading agents will be of different degrees. Immunological memory. Immunological memory is a very important and unique characteristic of adaptive immunity, by the virtue of which the immune system remembers the invading agent for a long period of time. If any foreign substance has entered the body once, the cells of immune system are primed against it and keep a backup of good number of cells which can recognize this agent. If this substance enters the body again, the response against it is faster than the earlier one. Both AMI and CMI store some cells as memory cells. Self and non-self recognition. Another important feature of adaptive immunity is clear differentiation between self and non-self structures. Components of adaptive immunity, B cells, recognize the invading agents by antibodies present on them as surface receptors, and T cells identify them through T cell receptors present on them. These cells do not mount immune response against self structures. The B and T cells possessing receptors able to bind to self structures are not allowed to mature in the primary lymphoid organs. Therefore, a state of tolerance exists for all the structures present on host's own body, and immune response can be generated against any macromolecular structure besides these. Mechanism for generation of adaptive immunity. Adaptive immunity can be generated inside a host by specific host defense mechanism, also known as third line of host defense, which functions in the body by two methods: humoral immunity or antibody mediated immunity and cell mediated immunity. Humoral immunity or antibody mediated immunity. Humoral is derived from the word humor, which means liquid, and humoral immunity means the immunity present in the body fluids. Body fluids such as blood, lymph and tissue fluids contain antibodies, complements and antimicrobial peptides. Major component of immunity in body fluids is antibodies. Therefore, it is also termed as antibody mediated immunity. Antibodies are Y-shaped glycoproteins, also known as immunoglobulins, and are produced by B cells. They are produced in response to the antigenic stimulus. B cells are produced from hematopoietic stem cells in the bone marrow. They are of myeloid origin. After they complete their primary maturation in bone marrow, they enter the main circulation in the body. They carry surface receptors which can identify the foreign invading pathogens. These surface receptors attach to the antigens present on the pathogens. This in turn stimulates the B cells for proliferation. Large number of copies or clones of this specific B cell are formed. Some of these are stored as memory cells and others differentiate into plasma cells. The plasma cells are tuned for oversynthesis of proteins. They produce antibodies in large number and secrete it in soluble form. This mechanism of antibody production is known as clonal selection theory. These antibodies circulate in the serum and attach to the corresponding antigens whenever they come across it. These antigens are cleared off from the body after the formation of immune complex. Cell mediated immunity. As the name suggests, this immunity works through cells. All the subsets of T cells are involved in cell mediated immunity. T cells originate from bone marrow but undergo primary maturation in thymus. Therefore, they are known as T cells. Following subsets of T cells are usually found functioning for cell mediated immune response. Cytotoxic T cells denoted by TC, helper T cells denoted by TH, regulatory T cells also known as suppressor T cells denoted by TS, cytotoxic T cells. T cells can be stimulated to exhibit the response by presenting foreign peptides on self MHC. All the nucleated cells of the body possess class 1 MHC on their surface to exhibit the antigenic peptides of the pathogens present inside them. These portions of peptides are produced inside the infected cells, therefore denoted as endogenous antigens. This is a method to report about intracellular parasitism to the components of cell mediated immunity. The T cells which have not come across with a foreign antigen are known as naive T cells, and after sensitization they proliferate and differentiate into T cytotoxic cells. All the circulating T cells possess T cell receptors. These receptors bind to the foreign peptides attached to self MHC, and the T cells get activated in turn. The sensitized T cytotoxic cells induce apoptosis in cancer cells or intracellularly infected cells. The self cell is destroyed in this mechanism, but this sacrifice protects the host from the hazardous consequences of spreading infection. Helper T cells. T cells work for humoral immunity as well as cell mediated immunity. T helper cells help in inducing the activity of B cells as well as T cells. Certain antigens require the help of T helper cells for inducing the B cells for antibody production. They are known as T cell dependent antigens. These antigens may be present in tissue fluids, but not inside any cell, therefore, also known as exogenous antigens. They are engulfed and presented by the antigen presenting cells such as dendritic cells or macrophages for presentation through class II MHC to the T helper cells. On activation, T helper cells stimulate B cells for antibody production. Regulatory T cells. These cells are known to regulate the activity of T cells. They are known to suppress the immune response if self surfaces are recognized. Therefore, it prevents autoimmunity. T cell types are morphologically similar, but they can be differentiated on the basis of surface markers. The cell surface markers which differentiate T cells are known as cluster of differentiation and are denoted by CD. CD follows a number which indicates different markers such as CD8 for T cytotoxic cells and CD4 for T helper cells. Types of adaptive or acquired immunity. Adaptive immunity can be acquired in the host's lifetime by acquiring the components responsible for it. On this basis, we can classify acquired immunity into two types: active immunity and passive immunity. Active immunity. Active immunity develops because of the response of antigenic stimulus. It is termed as active because it includes the active involvement of host's immune machinery. The host's immune machinery recruits AMI or CMI for developing the acquired immunity, which is a part of specific host defense mechanism. As a result, antibodies or immunologically active cells are produced for preventing the spread of infectious agents. Active immunity has an incubation period where, after receiving stimulus, cells of immune system undergo secondary maturation to perform effector functions. This incubation period is larger in case of first encounter with the antigen. This is primary immune response. The length of incubation period is reduced when the host comes across the same antigen for the second time. This is because of the immunological memory which was created after the first exposure. This is known as anamnestic or secondary immune response. Mechanism working behind this response is specific host defense mechanism, also known as third line of host defense mechanism, which includes humoral immunity or antibody mediated immunity and cell mediated immunity, which we have already discussed earlier. Passive immunity. Passive immunity can be provided to the host by administering pre-existing antibodies. Host's immune machinery does not participate in providing immunity because there is no antigenic stimulus. The antibodies are raised in some other individual to provide immunity to other recipients. These antibodies provide immunity immediately after its entry in the host. There is no latent or incubation period. This is a short-lived immunity and is provided at the time of emergency. It usually lasts for a few days to weeks. This works efficiently in case where the host's immune machinery is incapable of eliciting the response or there is no scope for waiting till the incubation period. There is no immunological memory in this case. Moreover, its effectiveness reduces on repetition. As the antibodies were raised in another individual and sometimes another species, the recipient's immune system may elicit immune response against this and clear it off from the body. Administering these antibodies for the second time leads to their faster clearance because of previous exposure. Therefore, it is less effective in comparison to active immunity and is employed only when immediate treatment is required. Natural immunity. When a host attains acquired immunity naturally, it is known as natural immunity. This can be active as well as passive, that is, it can be generated by antigenic stimulus or preformed antibodies also. Artificial immunity. When a host is immunized artificially to develop protection against diseases, it is known as artificial immunity. Artificial immunity can be developed in the host through active as well as passive mechanism. This can be clearly understood by going through naturally acquired active immunity, naturally acquired passive immunity, artificially acquired active immunity and artificially acquired passive immunity. First, naturally acquired active immunity. When a body comes in contact with a pathogen because of an infection, it recognizes the pathogen and mounts an immune response against it. This is known as naturally acquired immunity. When a pathogen infects the host, its immune system experiences the pathogen's antigenic structure, which leads to sensitization of T cells and B cells. The infection may be clinical or apparent, but the immune system gets stimulated due to the antigens present on it. As a result of the antigenic stimulus, host's body spends energy in building up the response against it. Thus, it is active immunity. Therefore, in this case, as the disease has occurred naturally and it has led to the introduction of antigen inside the host to generate immune response, so we call it naturally acquired active immunity. This is a long lasting immunity which protects the host from the same infections for years together. Naturally acquired passive immunity. Passive immunity is provided by introducing preformed antibodies. The antibodies formed inside mother are transferred to the fetus through placenta during gestation period and through colostrum after birth. The maternal antibodies protects the baby from infectious diseases during the developing stages of young one's immune system. The antibodies crossing placenta are IgG type and those transferred through colostrum are IgA type and resistant to intestinal digestion. Infants are able to respond immunologically efficiently only after 3 months of age. Till then, they are protected by maternal antibodies. This immunity is due to introduction of preformed antibodies, therefore, also known as passive immunization. The infant receives passive immunization against all the common infections from mother. Immunization of mother during pregnancy by vaccines further enhances the quality of passive immunization. the fetus or the baby is not spending any energy to build up immunity and is getting preformed antibodies. This transfer of antibodies is a natural process. Therefore, immunity acquired by this method is known as naturally acquired passive immunity. Next, artificially acquired active immunity. A host can acquire artificial immunity by vaccination, where the weakened infectious agent is purposely introduced inside the body to initiate the immune response against it in order to provide immunity. Vaccines are killed or attenuated preparations of microorganisms used to generate immune response in an individual. The host is experiencing antigen and building up immune response in the form of antibodies or sensitized T cells. It is known as artificially acquired active immunity. The vaccine contains weakened infectious agents or their products for immunizing the host. As the immunization is through antigenic stimulus, it is known as active immunization. Vaccination is done for the infants to protect them from infectious agents. They are routinely immunized against polio, tuberculosis, hepatitis, diphtheria, pertussis, tetanus, measles, and Haemophilus influenzae infections. Immunity provided by vaccines lasts for several years. Artificially acquired passive immunity. In certain conditions, patients are administered preformed antibodies to neutralize the effects of infectious agents. This can be used as a prophylactic step or for immediate treatment against some invading agent. Preparations of hyperimmune sera from animals or humans, convalescent sera and pooled human gamma globulin are used for transferring passive immunity. Some cases such as dog bite may lead to rabies or external tissue injury may lead to tetanus. These cases require instant clearance of the pathogens from the body. Antibodies which are raised in another host by inoculating them with inactivated rabies virus or tetanus toxoid are used for immediate treatment. Antibodies are raised in horse against tetanus toxoid and used as anti-tetanus serum ATS. This kind of immunity is provided artificially without the expenditure of host's energy. Therefore, it is known as artificially acquired passive immunity. Artificial passive immunization is also employed sometimes for suppression of unwanted active immunity, as in case of Rh incompatibility. If an Rh negative mother bears an Rh positive fetus, she gets sensitized to Rh positive antigens and produces anti-Rh antibodies. This may not cause harm to the first progeny, but can definitely abort the second progeny if it is also Rh positive. Therefore, if an Rh negative mother carries an Rh positive fetus for the second time, she must be given anti-Rh antibodies. These antibodies will neutralize the Rh antibodies and there will be no harm to the Rh positive fetus. Conclusion: body fluids and secretions have ample number of antibodies to combat with foreign antigens, whereas tissues and organs possess MHC for facilitating the recruitment of immune cells. These mechanisms of adaptive immunity provide surveillance in each and every part of the host to keep the body healthy.